The cancer stem cell hypothesis and progenitor gene rearrangements. Tumors are functionally heterogeneous and hierarchical. They are composed of cells that can initiate tumors, tumor initiating cells or cancer stem cells or CSC, and cells that arise from CSCs but cannot initiate tumors. The frequency of CSCs in a tumor is highly variable, often low. Cancer stem cells can, in many cases, are prospectively identified. Cancer stem cells may have different sensitivities to radiation or chemotherapy. Therefore, the concept of CSCs has significant clinical implications. When we look at this figure, we could see hepatopoietic stem cell undergo a transformational event which create pre-cancerous cells. Later, maintenance of self-renewal capacity with the help of that and additional transformational event that will turn to cancer stem cells. But at the same time, this hematopoietic stem cell transform to normal progenitor cells, which once again transform to mature differentiated cells. At the same time, that normal progenitor cell undergoes some transformational event to create transformed progenitor cells. With the help of this transformational events, then activation of self renewal capacity will be there, so they create cancer stem cells. Two hypothetical theories of cancer initiation and progression is very important. A. All subtypes of tumor are derived from the similar cells or progenitor cells. Different tumor phenotypes are then determined by subtype specific transforming events. Each tumor subtype is initiated from a single cell type, stem cell, progenitor cell, or differentiated cell. Random mutations can gradually accumulate in any breast cells, leading to the transformation into tumor cells when the adequate number of mutations has accumulated. All subtypes of tumor are derived from the similar cells or progenitor cells. Different tumor phenotypes are then determined by subtype specific transforming events. But next hypothesis states that each tumor subtype is initiated from a single cell type, stem cell, progenitor cell, or differentiated cell. Random mutations can gradually accumulate in any breast cells, leading to their transformation into tumor cells when an adequate number of mutations have accumulated. So, cancer stem cell specific therapy, tumor regression will be happening, but conventional gene therapy or conventional cancer therapy, that stem cell is there without any disturbance, so tumor relapse will be happening. Then the notion of cancer stem cells. Stem cells are essential for the initiation and long-term maintenance of the tumor. Self-renewing stem cells, transit amplifying cells are there, much of the tumor cell population, responsible for the expansion of the tumor, incapable of long-term maintenance of the tumor, perhaps post-mitotic in some cases. In others, further proliferation is possible, incapable of maintaining the tumor. Okay. Then, cancer stem cells and the problem of metastasis. Evolution of metastatic cancer stem cell. A tumor progresses genetic and epigenetic mechanisms and result in the emergence of a self-renewing metastasis that express different cell surface markers from the CSC that has derived tumorigenesis. Through a series of invasive processes that characterize metastasis, enters the bloodstream and seeds a secondary tumor in a distinct organ. Tumor cells express the chemokine receptors in the primary tumor prior to dissemination. Extracellular matrix is there, normal breast cells, malignant breast cells are there, lung metastasis will be taking place. Metastasis, chemokinases, kine and receptor. Therapeutic utility is limited because dissemination has already occurred at the time of diagnosis. Progenitor cell, differentiated cells are created, then epigenetic changes will be taking place. So the expanded or epigenetically altered progenitor cell pool, with that ONC, TSG, GKM, etc. are altered, benign tumor is created, epigenetic and genetic plasticity. Then primary cancer, epigenetic and genetic plasticity, invasion, metastasis, and drug resistance. The epigenetic progenitor model of cancer. According to this model, cancer arises in 3 steps. First, in an epigenetic alteration of stem or progenitor cells within a given tissue, which is mediated by aberrant regulation of tumor progenitor cells or TPG. The alteration can be due to events within the stem cell themselves, the influence of the stromal compartment, or environmental damage or injury. Second is a gatekeeper mutation, tumor suppressor gene in solid tumors, and rearrangement of oncogene in leukemia and lymphoma. Although these GKMs are themselves monoclonal, they expanded or altered progenitor compartment separately arising tumors. Third is genetic and epigenetic instability, which leads to increased tumor evolution. Note that many of the properties of advanced tumors like invasion, metastasis, and drug resistance are inherent properties of the progenitor cells that give rise to the primary tumor and do not require other mutations, highlighting the importance of epigenetic factors in tumor progression. As with other stages of carcinogenesis, metastasis is genetically controlled with the involvement of both enhancing and suppressing modifiers. Cellular phenotypes modulated by IGF-1, growth, apoptosis, invasion, metastasis, angiogenesis, and response to chemotherapy. IGF-1, IGFR, as positive regulators of metastasis. Mutant IGFR soluble receptor blocks metastasis but not tumor growth or breast cancer cells. Serum IGF-1 levels influence metastasis of colon cells. Cancer cell normal cell interactions, endothelial cells are there. And this fibroblasts transforms to endothelial cells. Cancer cells are also producing some other growth factors. Then immune cells are also there. Cancer tend to corrupt surrounding environment. Growth factors, proliferation, invasive matrix, fibroblasts, adipocytes, blood vessels, cytokines and proteases. Cytokines, proteases, migration and invasion. Tumor necrosis is a bad prognostic indicator. Tumor necrosis is a bad prognostic indicator. Hypoxia induces angiogenesis and promotes invasion. Hypoxia, invading cancer cell, and HIFs, VEGF, motility, invasion, metastasis, stroma, new blood vessel. Pathogenic defects in signal pathways extend into the tumor host interface. Cross regulation and exchange of enzymes, substrates, cytokines, and mitogens stimulates motility, invasion, survival, and growth of tumor and host populations. Locally and tightly regulated at the leading edge of the invading cancer cell. Cross-regulation and exchange of enzymes, substrates, cytokines, and mitogens stimulate motility, invasion, survival, and growth of tumor and host populations, locally and tightly regulated at the leading edge of the invading cancer cell. Next, cancer therapy, immune therapy, and gene therapy. Biologic therapy, immunotherapy, cytokine cellular therapy, tumor vaccine, hepatopoietic growth factors, and gene therapy. How this is done? The structure of antibodies. Then the IgG molecule. Fab arm waving, Fab elbow bend, Fab rotation, and FC wagging. Antigen binding sites, complementary determining region, antigen binding sites, complementary determining region, variable antigen binding region, light chain, heavy chain, variable antigen binding region, light chain, constant region, heavy chain, the antibody molecule. Look at the structure. Then chemotherapy, shortcomings, systematic Systematic nature of cytotoxin. Agents lack intrinsic anti-tumor selectivity, anti-proliferative mechanism on cells in cycle rather than specific toxicity directed towards particular cancer. Systematic nature of cytotoxin. Systematic nature of cytotoxin. Agents lack intrinsic anti-tumor selectivity, anti-proliferative mechanism on cells in cycle rather than specific toxicity directed towards particular cancer cell. Host toxicity. Treatment discontinued. Most of them had bad side effects such as no appetite, vomiting, loss of hair, etc. Immunotherapy of cancer. Potentially highly tumor specific. Can be effective against metastasis. Disadvantages. Can involve severe sudden onset life threatening. Treatment limiting side reactions. Limited by tumor heterogeneity, selection for unresponsive variants, and emergence of immune escape. Then monoclonal antibodies for cancer treatment. 3 mechanisms that could be responsible for the cancer treatment. Monoclonal antibodies act directly when binding to a cancer-specific antigens and induce immunological response to cancer cells, such as inducing cancer cell apoptosis, inhibiting growth, or interfering with a key function. Monoclonal antibodies are modified for delivery of a toxin, radioisotope, cytokine, or other active conjugates. It is also possible to design bispecific antibodies that can bind with their Fab regions both to target antigen and to a conjugate or effector cell. Binding to a cancer-specific antigen and induce immunobiological response to cancer cells, such as inducing cancer cell apoptosis, inhibiting growth, or interfering with a key function. MABs was modified for delivery of a toxin, radioisotope, cytokine, or other active conjugates. It is also possible to design bispecific antibodies that can bind with their Fab regions both to target antigen and to a conjugate or effector cell. Delivery of cytotoxic agents. Physically link antibodies to toxic substance for delivery. Radio immunoconjugates aim of delivering radiation direct to the tumor. Toxin immunoconjugates deliver toxins intracellularly. Antibody directed enzyme pro-drug therapy or adapt localize enzymes to tumor cell surface. Antibody directed enzyme pro-drug therapy. Antibody directed enzyme pro-drug therapy. Toxin immunoconjugates. Cell surface antigen must internalize upon MAB binding. When drug is released, it interferes with protein synthesis to induce apoptosis. 3 methods to attach immunotoxic drug to variable regions of MAB. Couple drug to lysine moieties in the MAB. Generation of aldehyde groups by oxidizing the carbohydrate region and subsequent reaction with amino-containing drugs or drug derivatives. Couple drugs to sulfhydryl groups by selectively reducing the interchain disulfides near the Fc region of the MAB. Direct attachment of MAB to drug by SS bonding. MABs. Then adapt enzymes. Antibody directed enzyme pro-drug therapy. Antibody directed enzyme pro-drug therapy. Chemically link the MAB to the enzyme of interest. Can also be a fusion protein produced recombinantly with the antibody variable region genes and the genes coding the enzyme. Chemically link the MAB to the enzyme of interest. Can also be a fusion protein produced recombinantly with the antibody variable region genes and the genes coding the enzyme. Convert subsequently administered anti-cancer pro-drug into active anti-tumor agents. Upon binding to targeted enzymes, this is converted into active drug. chemically link the MAB to the enzyme of interest. Can also be a fusion protein produced recombinantly with the antibody variable region genes and the genes coding the enzyme. Convert subsequently administered anti-cancer prodrugs into active anti-tumor agents. Upon binding to targeted enzyme, it is converted to active drug. Adapt enzymes. Targeted enzymes for prodrug activation. MAB enzyme binds to cell surface antigens. Enzyme converts producing drug enters cell resulting in cell death. Anti-growth factor MAB therapy. Angiogenesis, formation of nascent blood vessels. VEGF, one of the most upregulated antigens in cancer, protect endothelial cells from apoptosis by activation of PKC pathways and upregulation of anti-apoptotic proteins such as BCL2, actively mediated by tyrosine kinase receptors. VEGFR1 and VEGFR2 functions indirectly as surface factor or for tumor cells. Inhibit VEGF signaling, block the receptor using MAB, inhibit tumor growth and metastasis, deprives tumors of nutrient providing blood vessels. Then MAB's treatment for cancer cells. Naked MAB, radioimmunoconjugate, immunocytokine, immunotoxin, immunoliposome, cellular immunoconjugates. ADAPT, antibody directed enzyme prodrug therapy. ADCC, antibody dependent cell mediated cytotoxicity. CDC, complement dependent cytotoxicity. MAB, monoclonal antibody. SCFV, single chain FV fragment. Cancer gene therapy, direct attack on tumor cells, transfer of tumor suppressor gene, inhibition of oncogenes, suicide gene therapy. Oncolytic viruses, replication competent viruses, harnessing immune response to tumor antigens, chemoprotection, anti-antigenic therapy. There are two types of gene therapy: somatic cell gene therapy and germline therapy. Gene transfer is a new treatment modality and that introduces new genes into a cancer cell or the surrounding tissue to cause cell death or slow the growth of the cancer. This treatment technique is very flexible and a wide range of genes and vectors are used to clinical trials with successful outcomes. Gene therapy of cancer, potentially highly tumor specific, may have great value in combined modality approaches. Accessibility of cancer target is a major obstacle for general application. Then potentially dangerous side reactions from viral vector delivery agents. Antisense gene therapy, DNA transcription, mRNA, antisense drug, oligonucleotide, translation, protein. Antisense technology was emerged as an exciting and promising strategy of cancer therapy. The principle of this technology is the sequence-specific binding of an antisense oligonucleotide to target mRNA, resulting in the prevention of gene translation. So, suicide gene therapy, viral vector, enzyme encoding gene, tumor cell enzyme, prodrug. Then toxin spreads to neighbors. Suicide gene therapy is a therapeutic strategy in which cell suicide inducing transgenes are introduced into cancer cells. Suicide genes convert a prodrug to toxic metabolite. For example, the herpes simplex virus thymidine kinase gene stabilizes cells to the cytotoxic effects of specific drugs including ganciclovir and acyclovir. Enzyme producing combinations for suicide gene therapy given. Then generally modified tumor vaccines. Immune system may play a role in controlling tumor growth and development. However, antigens present on tumor cells are not sufficient to boost immune response. As a result, modification of tumor cells overexpressing certain genes may stimulate immune system to respond to tumor cells. Isolate tumor cells from a patient, transduce such cells with vector, retroviral vector harboring cytokine genes, inject such modified cells into patients. Strategy of genetically modified tumor vaccines. Strategy of genetically modified tumor vaccines: isolate tumor cells from a patient, transduce such cells with vector, retroviral vector harboring cytokine genes, inject such modified cells into patients. Next, virotherapy using viruses to treat cancers. Virotherapy with transductional targeting, adenovirus engineered to bind to and infect only cancer cells, does not infect normal cells, and adenovirus multiplies in cancer cells. Cancer cell burst and disperse virus to infect other cancer cells by lysis. Then virotherapy with transcriptional targeting. Cancer cells burst and disperse virus to infect other cancer cells. So adenovirus engineered to replicate under control of tumor promoter genes. Virus replicates only in cancer cells that have the tumor specific promoters. Cancer cells burst and disperse viruses to infect other cancer cells by lysis. Then disadvantages: lethal immune response in persons sensitized to adenovirus vector. Then reversal of genetic modification lead to lethal effects. Virotherapy with transductional targeting. Viral outer coat proteins, adapter molecules on engineered adenovirus, normal adenovirus receptor, no infection or cell killing, normal cell, but cancer cell receptor made only by tumor cells. Targeted virus takes over cancer cell, making so many copies of itself that it kills the infected cell. That's happening here. Receptor made only by tumor cells. Cell bursts and virus infects and kills other cancer cells. That is virotherapy with transcriptional targeting.